Thanks.
Well, now's as good a time as any, Mr. Miller. Take her down to periscope depth.
Yes, sir. Clear the bridge. Dive.
Stand right.
I'd like to apologies. I had my microphone muted. So I'd like to thank the audience for setting me straight what I totally do with you guys. I might have done a half-hour lecture being totally muted. So God bless you guys. I don't know what I'd do without you. I'd like to extend a very Merry Christmas as we celebrate the birth of our Lord and Savior Jesus Christ, who is the intelligent designer and creator. Tonight's topic is what I call a nerd night show. It's determining the germline mutation rates with somatic cells. This is pretty relevant to the question of young Earth creationism and young life and mitochondrial Eve. There's been an ongoing dispute between Dr. Dan Stern-Cardinal and Dr. Nathaniel Jeanson. And this is probably the first time I'm going to officially weigh in and just kind of give my opinions from what I've discerned in the peer-reviewed literature. So let me start sharing my screens here. And we'll get right to the topic, and then I'll have a little diversion and then get right back to it. You'll see what I mean. So let me start sharing my screen. The topic of mitochondrial Eve comes up a lot. If the clocks are what the creationists claim, then this would be very consistent with young Earth creationism and the genealogy of Jesus Christ. Let me just welcome and acknowledge my friends by name. Doki Doki Bible Club is here. Jeremy Martin, Flip Phone, Brandon, and if I missed anyone, apologies. Brandon asks if this is a new camera. Yes, I'm testing it out. I'm trying to see if this camera can actually work with my computer. So any feedback on whether you like the old camera or the new camera, that would be appreciated. So Nathaniel Jeanson has done a lot of work on mitochondrial Eve, both for humans and then the mitochondrial genomes of other creatures. And he has argued that life is indicated to be young, consistent with a literal reading of Christ's genealogy. And there's no surprise, Dr. Dan Stern-Cardinal disputes the account and has questioned the methods that in the papers that Dr. Jeanson has cited. So, welcome, BaptistChad. So, for a long time, and I don't know whether the dispute has been settled, and what I mean by settled is one party will say, you know, I was totally wrong, the other party was right. So I don't know if that's the case. But some months ago this summer, after I had debated Dr. Dan, he appeared on an open mic on Standing for Truth's channel, and I did try to get a hold of Standing for Truth and John Maddox, but I guess they're tied up. I'd presume Standing for Truth, that Mrs. Standing for Truth is insisting that Standing for Truth spent his Saturday night with her and the family, and that's perfectly fine. I was very eager to discuss this topic. So Standing for Truth and John Maddox, if you're watching us through recording, thank you very much for giving me your audience. I expect this discussion to be rather short. I'm hoping it'll be less than 30 minutes, less than 45 minutes. And Brandon asked, will you open an open mic? Tell you what, why don't we, would you like to join now? I would love some co-hosts. What do you guys think, huh? It's going to be a little interesting how I can do this. Let me go through my talk, and then we can do an open mic afterward, and we can all hang out. How does that sound? So just so just let me know. So I'll try to be quick. So I was going back to an open mic that happened sometime this past summer in 2020, and Dan was just kind of on a, Dan and John Maddox were just kind of rampaging on their views, and SFT was in there too. And I kind of weighed in and I said, you know, I'm kind of, obviously I side with Dr. Jeanson. I'm sympathetic to his view, but I would like to have more information from the peer-reviewed literature. Now, a little story about me and Dr. Jeanson. I've known about his work. I remember walking to a church one day and I saw this, I think it was from the Institute of Creation Research when he still worked for the ICR. And I saw his picture on the on this magazine, and I read about it, and I said, Lord, you really just encouraged my soul, because a PhD from Harvard is devoting his life to the study of creationism. I just praise God for that, and my heart was lifted. So that was my first encounter. I didn't even know about Dr. Jeanson until then. Then in 2017, he and I go down to Lipscomb University at a conference, at a meeting hosted by Dr. Joe DeWeese, who's also a creationist. And so I was going to give my presentation on the ENCODE project, Dr. Jeanson on mitochondrial DNA and speciation and dating of the genome. And I'll tell you, it was shocking. This is a Christian university, and there was some of the biology faculty there. There were some of the theology faculty there. I was horrified. I just gave a talk on ENCODE. I said, I do work for Dr. John Sanford. And they just went ballistic. They were hostile. They were practically screaming at me and Dr. Jeanson. And I was like, oh, this is supposed to be a Christian college? And we were kind of alarmed by that. And I said, yeah, this is nothing new. And I want to thank God for the creationists there and the ID proponents. They've slowly won the battle in battlegrounds like that. So at Christian schools, Christian seminaries, we are fighting the battle for creationism to be held in better esteem. And yes, it is very disappointing that in Christian schools, creationists are treated that way. I felt that people are free to disagree with us, and that's something I welcome. But raising their voices at us and ridiculing fellow brothers in Christ, that really wasn't cool. And that left a very bitter taste in my mouth. And I realized just how big a battle we have to fight in the churches, in the Christian schools, and in the seminaries. And so I'd like to thank Dr. Jeanson for being a fellow soldier in this fight. And he's an inspiration to me, he and Dr. Joe DeWeese. So now, Dr. Jeanson, after I gave my talk on the ENCODE project, and I may have mentioned the 4D nucleome and chromatin modeling, Dr. Jeanson gave his talk, and I have to tell you, it's not like I don't know any biology, but I couldn't understand a thing he said. And I said, Nathaniel, can you sit down with me for 2 hours and explain through things? And he did, and I thanked him. I understood a little better. I met him again I met Dr. Jeanson again. We met again at the International Conference for Creationism in 2018, just a year later. And I said, Dr. Jeanson, can we hang out and I'll have dinner with you and you could explain this mitochondrial clocking and rates? And we had a 1.5 hour really tough conversation. And I said, Dr. Jeanson, I have some background in population genetics, but this is way over my head. And let me think on it. So as much as I would love to endorse every creationist's work, it's just in my nature to want to be thorough. And I feel that I could do service to my fellow creationists by being able to articulate and defend their claims competently, and to be able to do it in front of biology faculty. And I have to tell you, it's pretty intimidating, and I've done this before, to stand up in a room full of postdoctoral students, that is, students that already have their PhD, and they're going back for even more. It's pretty intimidating to go up in a crowd like that. It's different than being on the internet, because in a crowd like that, you know everyone is really smart. And if you say something that's just a little off, they'll pounce on it. And even when you say stuff that's right, and I've done that occasionally, they'll just make snide remarks and just have to deal with it. So anyway, I had a session with Dr. Jeanson in late July, early August, and just to talk about the mitochondrial clocks. And I said, Dr. Jeanson, I'd like to be able to defend this, because this is going to be really awesome if we can show that mitochondrial E for humans and the corresponding mitochondrial quote unquote E for other animals is in line with a literal reading of the genealogy of Jesus Christ. This would be totally awesome. So enter Dr. Dan Stern Cardinal. We affectionately call him Dr. Dan. And Dr. Dan was saying the studies that Dr. Jeanson cited, they're lumping somatic mutations with germline mutations, that there may be 60 generations. I don't know that that's the figure that Dr. Dan cited, but that's a good estimate. That's about the Hayflick limit. The embryo will split and there might be 60 generations from that to a full adult human being. And Dr. Dan's claim or criticism, and correct me if I'm wrong, Dr. Dan, I don't mean to misrepresent you. I may misunderstand this, but to the best of my understanding, Dr. Dan's claim is that if we're sampling somatic mutations, that we can't use somatic cells to determine germline mutation rates. And therefore, Dr. Jeanson, is my understanding, Dr. Dan is claiming Dr. Jeanson is then being dishonest and disingenuous and bordering on fraud because the methods are all wrong. I'm going to dispute that tonight. I'm not claiming that I'm right, but I'm going to raise some issues that I've gotten from peer-reviewed scientific literature. So this isn't a creationism versus evolutionism debate. This is a debate just on materials and methods for inferring mitochondrial clocks. And I will argue that I think, despite misunderstanding what Dr. Jeanson has patiently tried to teach me, I think I finally get it. And I finally think Dr. Jeanson has a serious case of being right. And God bless you, Dr. Jeanson. I hope you're right. And if you're listening, I'm going to lay this out. So this is going to be a very nerdy night. Just a little digress, and I will digress a little bit here. Yes, it is funny talking about nerd topics on Saturday night, a little bit about my background. And it's a little scandalous. Oh, by the way, this is one of the papers in question by Parsons, a high observed substitution rate in the human mitochondrial region. This was the paper in April 1997 that has been followed up and by other studies. And it indicated, if we take the data to its logical conclusion, mitochondrial Eve, the mother of all females, lived only 6,500 years ago, very much consistent with a literal reading of Christ's genealogy. But let me digress for just a couple minutes. So what it's just by the way God ordained things that I'm doing this nerd thing, because many years ago, I used to love being in casinos and using my math skills to beat the game. So this is obviously a glamorized version of what it's like to be in a casino, because most people are not laughing. Oh, there's a joke. What's the difference between praying in a casino versus praying in church? When you pray in a casino, you really mean it. Anyway, this is one of my favorite casinos to play in, the Venetian. It's just so beautiful and elegant. And my other favorite place is the Bellagio. And that's the famous Bellagio fountains. And I got to tell you, since I love classical music, when they have these fountains going, they're playing Rachmaninoff's Rhapsody on the Theme of Paganini. And that's one of my favorite piano pieces. And I played it for church before. Just love it. So now, how did I get into this? Well, there is a famous professor from the Massachusetts Institute of Technology that figured out how to beat the blackjack game. And he used his math skills. He's a professor of mathematics, and he used high-speed computers in the 1950s or 60s. And he figured out how to beat the blackjack game. He actually published in the Proceedings of the National Academy of Sciences, which is amazing, how to beat the game. He went on to beat the game. The mafia almost killed him for figuring this out. And then he became a very successful hedge fund manager. By the way, when I'm playing blackjack, I have many tables like this memorized in my head. And so the idea is to walk into a casino, get to a blackjack table, and just play like you're a computer. Edward Thorpe, who I showed, he wrote the book Beat the Market, a Scientific Stock Market System. It actually worked till the stock market adjusted and said, we're not going to let you beat the market this way anymore. but Dr. Thorpe became a very successful Wall Street hedge fund manager. So he went from math professor to casino guy to stock market hedge fund manager. And he's featured in the book Fortune's Formula by William Poundstone. And so why am I telling you this? That's where I really wanted to go. That's where I really wanted to spend my Saturday nights, but the Lord had other plans for me. But incidentally, I'm friends with the Holy Rollers. They're actually called the church team. There's a documentary about this church team, and they're called the Holy Rollers in this DVD. And they're a bunch of Christians, pastors and elders that took the casinos for $3.5 million. And so I'm listed in the credits. So that's a total diversion. But I want to thank everyone who's joining me for Nerd Night, because now the Lord's placed me to do nerdy things on Saturday night. And so we're going to study DNA and ATCG. And I have to tell you, it's kind of scary when I'll read gene diagrams, gene listings and sequences, and I start to recognize motifs. One time I went into professor's office down there at Liberty University, and I said, hey, that's a restriction enzyme site, a palindrome. And so what do I mean? The diagrams I'll read will be like this. This is the genome, the mitochondrial genome of our mother Eve, the mother of all the living. This is the consensus sequence. And each and every one of us is a sequence that looks pretty much like that, minus a few changes. So now, this is the genome that is being disputed. One can go to this website called MitoMap and find this sequence. So yeah, this may look boring and nerdy, but this may be evidence that Eve lived only 6,500 years ago, and that this would affirm the genealogy of Christ. And this would also affirm, therefore, that the credibility of the Bible and its witnesses are trustworthy, and therefore Jesus is our Lord and Savior. So yes, if people are wondering what the relevance of this and why we should study it, I'm trying to explain that today. So going back to the dispute between Dr. Dan and I mean, not Dr. Dan and Dr. Jeanson, and I'm just trying to weigh in here. We can, and I'm sorry to get technical, and I think the people watching that are familiar with the technical details, and when we do an open mic, we can talk about this. If a parent, so what I did, let me see if I can get the MitoMap back. Sorry, wrong picture. Let me try to get this. Where's the MitoMap? Landscape. So all I did is I went to, like, say, line 841 here. Let me see if I could find it. I just took a arbitrary line out of this MitoMap of the mitochondrial DNA. And I cut and pasted it here and said, okay, let's just hypothetically say this is one mother. This is what her sequence looks like. And then the child has a sequence like this. Now, there's some subtleties to mitochondrial DNA. There are terms like heteroplasmy and homoplasmy. That's because cells can have anywhere between, I've heard numbers from 9 to even 600,000 mitochondria. And so that means even though there's a single nuclear genome, there could be 600,000 mitochondrial genomes in one cell. And that causes some complications in in sequencing and figuring all this out. So just suffice to say, we can talk more in detail about that. But what I just want to try to simplify things is if we have, say, a parent's mitochondrial DNA and it looks like this, and then the child has a mutation that looks like this, so the C gets turned to an A in their cell. What ends up happening is if we sample all the somatic cells in the child, we should see most of them have that same mutation because the cells kind of clone approximately their mitochondrial DNA across all the somatic lineages. So let's compare and contrast this to what Dr. Dan is claiming. And I can understand why I believe he made a mistake. If we only took one somatic cell and compared it with a mother and just counted all the mutations, yes, of course, that might not give a very accurate reading of the germline. So Dr. Dan is correct in that sense, but that's not what these researchers do. That is not at all what the researchers do, and I'm going to try to prove my point here. So Dr. Dan would be correct if the researchers did it the dumb way, which they don't do. So this is what's in question. What kind of sequencing technique is Dr. Dan appealing to? Is it some sort of mythical technique or the way they really do it in the lab? And that's a question that I had been wrestling with since I had that meeting with Dr. Jeanson in 2017 and also in 2018. And to pull an imitation of Erica, I'm not a geneticist. I'm not a geneticist. I do population genetics, but it's not sequencing genetics. There's specialties. There are people who do sequencing genetics. There are people who do population genetics. I'm more on the population genetics side, the theoretical. There's stuff with the sequencing that is a real specialty. So in that sense, I'm not a sequencing type geneticist. I'm more of a protein guy, if anything. And so I'm just going to claim to be like Erica. But that being said, I'm just going to speak my conscience and weigh in here. So what I think Dr. Dan is describing is something that no one does. He would be absolutely right if that's what they did in the lab, but I don't think that's what people are doing, and that's what I'm going to argue. Therefore, if we describe the actual lab procedures, materials, and methods, it would seem to me that Dr. Jeanson has a much better claim and that all the to being correct, and that all these accusations of him being disingenuous or a liar or a fraud using knowingly wrong data, I don't think they're founded. Now, that said, I could be wrong. I want to get the discussion going. So, going back to my diagram here, if we use phylogenetic methods and we see all the somatic cells have the same mutation, we can just sample the somatic cells, like say hepatocytes, erythrocytes, myocytes, you name it. Those are various cell types. And they all have that same mutation, even if they have, let me see if I could find that mitomap, even if they have other mutations elsewhere in this huge, not really huge, in this 16,000, roughly 16,500. See what, let me give you the exact figure. That's me in numbers, okay. 16,569 bases. So there's 16,569 places another mutation can land, but in that particular region, we're going to look for the persistence of that mutation. So yes, Dr. Dan is correct that there could be mutations accumulating all over the place here in other somatic lines, but that's not how we count germline mutations. And then the reason I'm coming out tonight is I found peer-reviewed articles that support my viewpoint. So just a little bit of a preliminary. This has some relevance to the math that was created by Sewell Wright and Ronald Fisher. The math in the wiki article on genetic drift is related to these questions in the form, the math in the form that we see it here. It's not directly applicable to the problem because this is the diploid model and all this fancy math. Don't get intimidated by it. Just suffice to say, and this is why I was getting headaches when I was talking to Dr. Jeanson. I said, I kind of understand some of this math here, and I don't see that. I'm trying to visualize what the equations are that would prove his point, because this is a very simplistic model. And this is where we have constant or shrinking populations. In the case of mitochondrial DNA going from an embryo to a full adult, these rules don't exactly apply. So we had a long conversation about that. But so I'm just mentioning that in passing. There is some involved math in this, but the basic thing that I just want you to get, we can figure out, I tried to lay out the difference between a germline mutation, which is the thing that we're going to calibrate the mitochondrial mutation rates across generations. That's the one that's of interest. We can deduce it even though we sample somatic cell lines. And I'm showing you the technique here. We can do this using what we call phylogenetic methods. And I know how to do a little bit of molecular phylogeny because I studied under a evolutionary biologist, a computational biologist, and a bioinformatician at the NIH. And one of them worked for Eugene Koonin at the NIH. So I learned some of these methods. So this is just straight phylogeny that we can build what is called a consensus sequence for the zygote. And when we can do that from the somatic cells and build a consensus sequence, we can make an estimate for the structure of the germline mitochondrial DNA. And from that, we can infer the mutation, mitochondrial germline mutation rate, which Dr. Dan is disputing. And I'm like, this doesn't look good for him. So I may have just laid all this out and I was like, yeah, I think Dr. Jeanson's right, but can I just get some peer reviewed papers to help me make my case? And here we go. Here's one paper, The Landscape of Germline and Somatic Mitochondrial DNA Mutations in Pediatric Malignancies. And so my 3-year journey, and they begin to describe their materials and methods, which I was very interested in. And we go to the materials and methods section. I'm just going to read this verbatim. I'm sorry, it's technical, but this is what I had to go through to understand I think Nathaniel Jeanson's right. So apologies to the reader, but I'm just going to read it verbatim. Let me try to make this maybe a little larger. Oh boy. From the materials and methods section. And I want you to focus on this word VarScan too. We'll revisit that. It said mitochondrial DNA, somatic and germline calls were made by VarScan 2 and further filtered out by our LUBA variant caller, which is a custom somatic variant caller that supports the pediatric genetic tests at our institution. VarScan 2 was run in somatic mode with standard parameters, which require at least 100 times coverage in both the tumor and normal samples, min coverage 100, at least 2.5% of alternative allele frequency in the tumor to make a call, min var frequency 0.025, and minimum base quality of 20 at position, min average qual 20, and strand bias filter, strand filter 1. We removed known recurrent false positives caused by misalignment in the poly-C region, and I'm just going to skip. And they just keep going on. And then they go back. I'm just going to move a little here. Our LUBA variant caller was run with the same parameters as VarScan 2, stringent strand bias filter 2.5%, AF, and minimum 100 times coverage, and was used to confirm calls by VarScan 2. LUBA supported 94% of tumor-only calls and 99% of germline calls. We determined this set of high-confidence calls and used them for downstream analysis. So this goes on and on about how they're able to infer mitochondrial mutations that happen specifically in the somatic line versus germline mutations. So I looked up this Luba filter, and unfortunately, this looks like such a specialized tool. It's not widely available. However, there is a paper, and oh boy, I think I lost the citation. If you all will indulge me, I'm going to try to bring it up. So let me think on the fly here. So VarScan, let me look for the VarScan. VarScan. So I'm going to go all the way up to the top. I'm going to look for VarScan, and I'm going to show you how they do this. VarScan. I had this VarScan thing. It was so awesome. So please forbear with me while I look this up. VarScan. Well, I'm going to go look in the, there it is. VarScan, variant detection and massively parallel sequencing. So there is this article on VarScan. And if one reads it, now it goes on, but one is able to use this tool to distinguish what are somatic mutations in the mitochondrial DNA versus germline mutations. And this is the tool. This was the breakthrough I was looking for, because intuitively I knew that I ought to be able to find a way to distinguish germline versus somatic line mutations. And so there are more papers. This one from the Annals of Oncology. And I need to point something out, because the first paper on mitochondrial that raised the issue of mitochondrial E being only 6,500 were done on blood cells. Now, why would we pick blood cells? I'm going to read from here. Okay, so it says in this article, and I'll give the citation, but let me read this. Somatic mitochondrial DNA mutation was defined. Okay, somatic mitochondrial DNA mutation was defined as the heteroplasmic mutations observed only in HCC or non-HCC tissues. That's either cancerous or non-cancerous, the particular cancer that they're studying, but not in blood samples in the patient. Considering that most of mitochondrial DNA mutations in blood samples may be germline. So apparently, for some reason, blood cells are awesome cells to test mitochondrial germline mutations. And that was exactly some of the papers that Dr. Jeanson was citing. And even in those citations, in those articles, it was saying that that they were able to distinguish mitochondrial germline mutations from mitochondrial somatic line mutations. Not only that, since blood samples were used, it tends to be a little bit more stable. It doesn't acquire as many somatic line mutations, and that was a concern. By the way, just for what it's worth, blood cells, what we call erythrocytes, I think that's the right term. Correct me, anyone, if I'm wrong. These erythrocytes don't have any nuclear DNA, which is really amazing. That's neither here nor there. I just thought that was kind of cool. So there are other articles that deal with this, and that's really my presentation. I just wanted to make my case. I think Dr. Jeanson's right, and it looks really favorable for the young Earth creationists. So now I'm going to try to, anyone who would like to join an open mic here and talk about what my findings. And I've delayed weighing into this because I didn't feel I had sufficient, a level of knowledge that I felt comfortable enough defending my friend and colleague, Dr. Nathaniel Jeanson. And now I really wanted to do my friend justice by going to the peer-reviewed literature. So if you'll give me a moment here, I actually have to send the invitation from one computer to another so I could post it on another computer. And let me see, do I have even permissions on my own website? Let me look. Please give me a moment here. I have to log out of one account. So this is going to take me a few minutes. This is a little bit annoying. And so I'm going to log in. And in the meantime, and I got to say, I want to be very careful about who I let into the open mic. I don't know if some of you were aware, but it was really kind of sad what happened to our dear friend John Maddox. Some troublemaker went there and just started showing things that he shouldn't be showing, and it got John Maddox in trouble. Also, there's certain people I'm going to have to just say you're not welcome to join the stream. This website and this channel is meant to serve a family-friendly audience, and so there are certain standards. And I want homeschool parents to be able to feel comfortable sending their kids here. There are a few times I've kind of like violated my own policy here, and I'm trying to figure that out. I'm trying to create channels where I could have a little bit more open conversation. So hang on here. I'm trying to log into my own YouTube channel so that I can give you the StreamYard link. And Doki, I really want to thank you. At some of my other live streams, you were so kind to keep track of the StreamYard and letting people in. So we might have a small crowd tonight, and we could talk about this. We can talk a little bit about peripheral issues, but I'm kind of running out of gas, and so I would appreciate if we could just focus on a little bit of what we talked tonight. So let me see. Okay, I found the right show, found the right stream, and I'm going to type it in, and please join. If I don't let you in, I'll try to find some way of letting you know that I see you in the StreamYard, but I let certain people in. That's just going to be my policy. All right, so please join. Oh, my goodness, I see Ian Chen here. So just gentlemen and ladies, please keep it family friendly. BaptistChad, welcome.
Shalom. Hello, Sal.
Shalom, and thank you for praying for my mother. God bless you.
Yeah, always, brother. I always got you in my prayers. If there is anything, let me know. I'll be handing a prayer to the Lord.
So, I think I hope this channel would be of interest to you. It's oriented toward. It was first oriented toward the homeschool crowd, was my vision, but I'm realizing at all levels of education, and not just homeschool, but public and private schools and universities, because I've been actually kind of horrified at how they even teach arithmetic in public schools.
Mm-hmm.
It's kind of scary.
Yeah, it's a mess in my public school.
I mean, I was like, I have a math degree, and I couldn't figure out how to do arithmetic that way that they teach it in the elementary schools. That really bothered me. So some of y'all that are out there and heard me saying that I'm making the channel for homeschoolers, I'm trying to widen it. So there's going to be this main channel that does evidence and reasons for the Christian faith. But then I'm going to have like daughter channels, one that's just kind of science, where we do basic math and physics and chemistry, and then even advanced all the way to the graduate level. I have a heart for kind of relearning general relativity.
Yeah, I like math. Math's definitely a nice subject. Well, maybe I could ask you this question: do you believe math is discovered, or it's we just Do you believe math we discover math, or we created math?
I think we discover it through what we observe in nature. Now, math, the correct term is, we're beginning to understand this. What we call mathematics is the study of relationships that are algorithmically compressible. There's a lot of things in the universe of numbers that are not computable. And we would have no mathematical equations. The universe is structured in such a way that a lot of its laws, a lot of the physical laws and things are accessible to human understanding. Because it could have been so complex we wouldn't have been able to make math laws.
Amen.
My professor of mathematical physics said nature, we were studying these differential equations. He said, there's no higher order than 2. 2 is the highest order for the laws of nature. And I found that really amazing because that makes it solvable. Otherwise, we'd have some really hard times if the major laws of physics, I mean, we'll see lots of things that are beyond second order differential equations in our analysis, but the statement of most of our basic laws, at most it's second order. So it seems God has structured the universe to be understandable.
Yeah.
Science is a gift. Science is a gift and miracle from God.
Amen.
And this is not just from Christians. If we read some people who are non-Christians, they've said the same and not in different words, but it's the same idea. Eugene Wigner, a Nobel Prize winner in physics, he said the unreasonable effectiveness of mathematics in the natural sciences, and he was struck how it just works so well because he could figure so many ways that nature could be constructed that we wouldn't be able to analyze it with mathematics. So to your question, I think it's discovered the way that the math of physics is discovered.
Yeah.
Helps the process along.
Yeah. I had my point of view, and just tell me what you think about it. I believe math is kind of created, God let us know. If we look at the sun, we will define the sun as being one sun when we watch at the sun in our universe. But if human weren't there, I would just define as it is there. Would that make sense?
Can you say that again?
Yeah. So if you look at the sun or look at the star, right? We just define it as being there, right? There's a star, there's a sun. But if we weren't to exist, we would just define the sun as being here, right? Would that make sense?
I didn't quite get your drift. Try one more time. I'll try to listen intently.
All right. Okay. So what example could I take? It's more simple. All right, let's just define the speed of light. The speed of sound or speed of light. We would just define it as being there. But as we understand it, we describe it as with numbers.
Yes.
But would you say the speed of sound and the speed of light is actually a mathematic that God shows in a schematic? It's actually represented by number in our universe?
No, we can measure it, and I mean you can measure the speed of a car, and we can measure the speed of sound. We can measure the speed of light near the surface of the earth, and we can measure the speed of light going through a vacuum and also through various materials. So there is I'd like to acknowledge some of the others who've joined us tonight. So there's Ian Chen. Welcome, Ian. Thank you for visiting. And Thamazo, and he says, "Hey, Sal, See, I ought to be able to show. I've never done this before. Ought to be able to do a show here. Said, hey Sal, I joined from our Reddit private messages together. This channel is amazing. There's literally no channel specializing in the intersection of theology and religion. So thank you very much for the kind words.
Yep.
I recognize the handle. My apologies. Maybe it was a different handle there. So what we do with the mathematics is, the reason the math is important is sometimes when we can't make a direct measurement, we can make an indirect measurement. So from the things that we know, we're able to deduce the things that we don't understand.
Mm-hmm.
And it's been very helpful because we can't really see the atom with regular wavelengths of light. We have to use machines, devices that will infer the structure and shape of atoms and molecules.
So, it's kind of amazing how everything's so complex. Just make me think about the Bible passage where, in the book of Isaiah, he looked at the heavens and he says, who is man mindful of? Who you created all of those, and the heavens really declares the glory of our God, and it's amazing.
So just over time, I hope you can let me know how I can be of service to you. I'd love to, one way for people to relearn or learn any subject is to try to teach it. So that's actually one reason that I'm very interested in teaching and making teaching videos, is partly so I could relearn and gain mastery of the subjects that I studied. And so if I could point this out, see my 5 degrees up there?
Yeah.
I've forgotten so much of what I learned. I need to relearn it. I'm so embarrassed. I was just trying to do some high school chemistry and physics and math, and I'm like, yeah, I've got a long way to go. I mean, I think I can relearn it, but it's going to be good to go back and
absolutely
some of the stuff. So that's why I'm teaching, and I hope it can be of service to you, because we can actually understand creationism better if we understand the science. So that being said, since we're talking about DNA, and let me share my screen here.
Yep. Yeah,
we'll look at the mitochondrial DNA, and since that was the topic of tonight, and let me just try to shuffle some screens here. So I'll bring up the mito map here, and then maybe we could have, if there are things that didn't quite register, I hope we can talk about it. So how familiar are you with cell biology and mitochondrial DNA is?
To be honest, I'm really a young kid, and I'm just here to listen and learn about you, because I'm really interested into the subjects of, evolution and creationists, and I would like to try to get a PhD in biology or physics. So yeah.
Oh, praise God. Praise God. Now, let me tell you, it's tough to go to a secular school, but I think that's the best place to go. And one reason that I have a passion for talking to kids your age is that there are some materials in creationism that I think don't serve the young well, because they're not well argued. And once these students go off to college and they are in an environment where they may be like 5%, maybe 10% of the Christians, and maybe they might be the only creationist in the class, say a biochemistry class or an evolutionary biology class or a bioinformatics class. It's not just that, but you will learn things in the classroom that it's like, well, why didn't I see any of this in any of my, like answers in Genesis or CMI dealing with it? Like, for example, once you start doing what they call molecular phylogenies, that's a very good argument for evolution. And I don't think hardly any creationist deals with it correctly. Very few. So
yeah,
I'm going to try to help you see that, because I am a little horrified with what I hear from some creationists saying, like, they deny the existence of the nested hierarchy. And it's like, no, it's pretty apparent, because I see it all the time when I'm gene browsing. So while you're here, why don't I try to give you through a free lesson?
Amazing, amazing.
All right. So, and Ian Chen said, Sal, you have to go to a secular school to get a PhD. Unfortunately, no creationist schools are accredited. Let me just put that there.
Yeah, and real quick, I think
and I have to tell you, I had an opportunity to study at the ICR grad school. I said, no, I'm going to a secular school. And I think if you're well equipped, and Dr. Jeanson's a good mentor to have because he was raised in a Christian home, a creationist home. And I asked him, how come you're still a creationist after going through school? He said, well, when I was growing up, his parents, he said, when he was growing up, his parents gave him both sides, taught him both sides of the argument, taught him evolutionary biology and creationism. So one way to be exposed to both sides is to study it when you're young. And then because if you go to college and you're relying on the stuff that you just see in these, like, say, one-page articles on answers in Genesis or CMI, it's just not going to cut it. Your faith could be shredded to pieces. And one reason I'm very careful when I try to say things, I said, I think this is correct. Maybe a part of it is faith, but I'm not going to argue that this is airtight. And that helps. Just when we're willing to say we don't know and we don't have all the proof. Oh, Ian Chen asked, so we'll give a lesson here. And thank you. I will try to do my best. And Ian, you are welcome to join the stream. Doki, if you could help out. So we'll do a lesson here. And let's see how efficiently we can walk through this. So the first thing is, we can have various definitions of life, but the nicest definition is cellular life. So let's just look at a diagram for a cell. And so I'm doing this totally ad lib here. I could totally mess this up. So let's look at a human cell. I think this is going to be a better diagram. Hopefully this is clear enough, and I know the text is really small. So this is a archetypal animal cell, and so that would mean a human cell has this model. So one of the evidences of evolution, in favor of evolution, even though I'm a creationist, we have to acknowledge that all animal cells have a lot of similarity to humans have a lot of similarity to other animal cells. We can call ourselves animals. Now, there's a verse in the Bible in Ecclesiastes that says, men are but beasts. I don't know the verse off the top of my head, but it's biblical to say that men are but beasts because it says that in the Bible. So there's an aspect of us that, our souls are special in God's eyes, but there's also, God gives us reminders that we're mortal. We're mortal, just like all the other creatures. We can't, that's God's way of humbling us. So all animals have cells that can be conceptually described this way. So the cell has a nucleus, and this is where the DNA and what we also call chromatin is stored. And then it has a membrane that kind of packages the cell so the stuff doesn't leak out and spill out. And then we have something called the mitochondrion. So there are actually two genomes, many genomes in the cell. We have the DNA that's in here, and then the DNA that's in like the mitochondrion. And you can see there are actually multiple mitochondria. Some cells have, I think, as little as 9. And unfortunately, I don't have a citation for that. But I've seen some citations that said some cells could have 600,000 of these things.
What?
So the DNA, you have DNA that's centralized here, and then DNA here. It's a different DNA in the mitochondria. So question. And by the way, this is what the DNA in the mitochondria looks like. That's what they call mitochondrial DNA. So go ahead and ask. I'll try to answer your question.
Yeah, yeah. My question was the little blue thing, mitochondria. Yeah, mitochondria. Sorry, because I speak French. I have a big accent. What exactly does it do in the cell?
That's a good question. And maybe so I don't mess this up, we can always go to Wikipedia, which is generally really good. So I don't want to mess this up. And I'll try to interpret it. Mitochondrion, plural is mitochondria, is a double membrane bound organelle found in most eukaryotic organisms. And I'll try to explain that. Eukaryotic organisms are things like animals and plants. Bacteria are not eukaryotic. They're what they call prokaryotic. So there's a big divide between the eukaryotes and the prokaryotes. These are just terms. You'll learn them. It's not hard, just like learning names. So the mitochondria is a double membrane bound organelle found in most eukaryotic organisms. Some cells in multicellular organisms lack mitochondria, mature mammalian red blood cells. Wow. A number of unicellular organ. Well, we don't need to go there. So let's look at the function. What you asked me what it does. The most prominent role of mitochondria are to produce energy, the energy currency of the cell, ATP. That's adenosine triphosphate. That's an acronym. Through respiration and to regulate cellular metabolism. I just say it's the powerhouse that generates all this ATP. It's a very important chemical to make the cell work. So it's generating ATP.
So it's basically doing the energy like the sun reflecting on the earth, giving energy. So basically, do the energy for the cell to produce.
Yes, it does something. It uses what they call. By the way, I'm giving you like junior level college here, so there'll be some gaps. Oxidative phosphorylation. I'll show you a little bit of this. I'll show you some videos. What it does, let's see, oh my goodness.
This looks so complex.
Yes, okay. I'm going to be teaching you biochemistry, but what I wanted to just say is you don't have to go to junior level college to be able to get the important things as far as learning how it's relevant to creationism, okay? Mm-hmm. So I'm just going to give you some of the organelles here. So inside the mitochondria, you have, let me see if I could find this. I'm going to give another diagram of the mitochondria. So we have so here's a good photo right here. So we have this cell, we have the nucleus, we have the mitochondria. In the mitochondria, if you zoom out, you have this, the cristae and the outer membrane and the inner membrane. The inner membrane is really important here. So now I'm going to focus on the inner membrane. In the inner membrane, you have this thing called the citric acid cycle that's converting kind of our nutrients, and then it's going to go through a process and create all this stuff called ATP. And the ATP is a very useful chemical to power life. So when we digest food, be it sugars or proteins or whatever, it eventually lands in a lot of the nutrients end up in a metabolic pathway, which is just a bunch of chemicals that take those chemicals and generate all these other chemicals, and then it works with a machine called ATP synthase to make ATP. Now there are various other pathways to make ATP, but this is the main thing that the mitochondria does. And I'm going to show you maybe I could show you how this ATP synthase works. Let's see if I could ATP synthase animation. See if I can give you an animation of how this machine works.
Oh
And
yeah,
this,
I've seen that on Dr. Rob's Rob channel. He has a good video on ATP synthesis. It's like literally pump like atom out of your body. Like it pump like I think it's like hair or something like that. It just pump it over. It pump oxygen, I think, and it just throw it out.
so why don't we just review this and just remember this particular ATP synthase is in the mitochondria. There's another thing in plants called the chloroplast. It also has an ATP synthase machine in it.
Yeah.
So let me just share this and just enjoy, and it'll give me a chance to get something, a little hot coffee. So let me show this here.
Yeah, you're not sharing your screen right now.
Yeah, I'll start to share it. Just
All
one
right.
moment. I wanted to get a Chrome tab.
You should rename yourself Professor Sal.
No, I'm nothing like. I mean, I'm just like a joker compared to Rob Stadler. Dr. Rob Stadler's just, oh my goodness. I'm nothing compared to some of my colleagues. I'm just a troublemaker on the internet. So okay, let me know if you're hearing the sound. So we'll try this.
ATP synthase is a molecular machine that works.
Can you hear it?
Yep.
Okay, let's play it, and I'm gonna just step out for a little bit. Enjoy. It's just
All
3
right.
minutes long.
ATP synthase is a molecular machine that works like a turbine to convert the energy stored in a proton gradient into chemical energy stored in the bond energy of ATP. The flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only protonated subunits can then rotate into the membrane, away from the static channel assembly. Once the protonated subunits have completed an almost full circle and have returned to the static subunits, an exit channel allows them to leave to the other side of the membrane. In this way, the energy stored in the proton gradient is converted into mechanical rotational energy. The rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPase, which catalyzes the formation of ATP. The F1 ATPase portion of ATP synthase has been crystallized. Its molecular structure shows that the position of the central shaft influences the conformation and arrangement of the surrounding subunits. It is these changes that drive the synthesis of ATP from ADP. In this animated model, different conformational states are lined up as a temporal sequence as they would occur during rotation of the central shaft. Like any enzyme, ATP synthase can work in either direction. If the concentration of ATP is high and the proton gradient low, ATP synthase will run in reverse, hydrolyzing ATP as it pumps protons across the membrane. To show the rotation of the central shaft, a short fluorescent actin filament was experimentally attached to it. Single filaments attached to single F1 ATPases can be visualized in the microscope. When ATP is added, the filament starts spinning, directly demonstrating the mechanical properties of this remarkable molecular machine.
Amazing, amazing. You're muted, Sal.
Oh, thank you.
Yep. Amazing video. So basically the ATP, so the red thing is producing proton, and proton go to the shift, and after produce ATP synthase from what I've seen.
Well, what I would suggest is going and watching, rewatching videos like that. And even myself, I have to watch it like 20 times to get all the parts right. And that's even after having studied this in school. And so a lot of the study of biochemistry is just like trying to memorize a phone book. There are all these parts and just listing what goes.
Taking notes too.
Oh boy, it's terrible. So let me try to see if I need to find another video. And Ian says, if intelligence created mitochondria, then isn't that evidence it can arise naturally? Because you've just shown the conditions that a process can thus form. And that's a good point. Let me just give what my view of this is that we can't make machines this sophisticated. I think that a miracle created the first ATP synthase machine. And as you study biology, just ask yourself this question, just keep an open mind. Can I build something this sophisticated? And then if you can't build something this sophisticated, ask if random mutation and accidents, a series of them, and natural selection can build it. It's very hard to have a living cell without an ATP synthase system. There may be some situations where it may not be needed hypothetically, but I'm not immediately aware of it. There are other metabolic pathways to create ATP, but this is a very special machine. And the more that I've studied biology, the more amazed I've been. So let me share my screen here. And so Ian, if you'll forgive me, I want to focus on BaptistChad here. I'm trying to just give him some basic biology. I'm not going to, I'll touch a little bit on creationism, obviously, but I'm going to try to focus on the basic science. So we saw that, we saw the ATP machine, and then that's inside, that's connected to the inner membrane of the mitochondria. And we also had a picture of the mitochondria like this. Let me see, the mitochondria like that. So you can see that little ATP synthase machine is in here somewhere. It's just amazing. So the inner membrane, so you have these ATP synthase machines, and there are other things that are all connected in here. And you asked about plants. The ATP synthase, see if I could, maybe I'll get blessed. Let's see, ATP synthase, mitochondria, chloroplast. In plants, we have other organelles, and see if they have, oh, here's the diagram I wanted. It's not a very good diagram. It's terrible and it's blurry. Let's see if I could find a better one. If I can't, it's just, nope. That's about the best one. And I'm sorry, it's so blurry. I can't believe it.
Yeah, you can try to zoom right there on it without opening a page.
Yeah, I don't know why it got so blurry. So let's do what you suggest.
Yeah,
I'll just zoom in.
Yeah.
And
yeah, I mean, I can see it fine like that.
Okay, so in the mitochondria, you see the ATP is pointed a certain way. The little machine head is inside. In plants that have chloroplasts, it's also another organelle. The ATP synthase is pointed outwardly. And so let me show you a plant cell. It's slightly different than an animal cell. Let's see if I could find a... So the plant cell has a lot of similarity to an animal cell, it has a nucleus, it has mitochondria, but then it also has these things called chloroplasts. And so we have the ATP synthase in both the mitochondria and the chloroplast, but they're oriented differently. Now I'm getting way ahead of myself. The real point is that what I'm trying to show is that you're asking what the mitochondria does, and I wanted to just say that you could see that in a human cell, there'll be multiple mitochondria, and in that mitochondria is also the DNA. So let me see if there, I doubt I'll find anything, but let's just try it. Mitochondria.
So the mitochondria is basically pumping because of the ATP synthesis. So it's pumping kind of energy out. So it's produced by the proton, and after it's by the shift, and it's kind of pumping stuff out and entering stuff too, from what I've seen. Is that right, Saul?
I'm embarrassed to say I don't know. I have to watch the video again. That'd be the right thing. And so I'm kind of embarrassed.
Oh, there's no problem. Don't worry.
That's why I'm so glad we have references like that. So I'm looking here. Oh, here it is.
This is really amazing. This is high design.
Okay, so we're talking about the ATP synthases. They're all lined over here, and you can't see it because they're so small on the inner membrane. And it says here, the DNA is located here in these little circular things. So I'm sorry. Let me get rid of that. Let me open this.
So
this is what I want. So you get the mitochondria here. You have the ATP synthase, but then you have these little ring things here. These little rings. That's the mitochondrial DNA. That little ring, and that's the focus of the talk tonight.
So sorry. Is that DNA here pumping the nucleus?
Huh?
So you know the nucleus is what produced the DNA?
The nucleus has most of the DNA, but not all of it. So let's go back to the diagram of the cell. Let's go back to the diagram of the cell. And this is why it can be confusing. Most of the DNA, actually, this is a good diagram. We have it right here. Most of our DNA is in, just to give a comparison, we have roughly 3.3 billion bases of DNA in the nucleus in general. Cells that have nuclear DNA. Not all cells have that, and that's another story. But in general, you have 3.3 billion bases here. But in the mitochondria, it has a little bit of DNA and only 16,569. And I'm showing you the spelling of that DNA right here.
So this is just basically code. Do we know what does that mean exactly? All those letters, does they just code for protein?
I don't know. A lot of them, there's a segment of this that will code for a protein. Yes. So let me see. Is DNA a code? The correct term, people will say that it's not the most exact language. DNA is used to implement a code. Mm-hmm. It's the hardware. It's kind of the materials that are used to make to implement a coding system. So that's just a fine point there. So this whole talk is then just to give you context in terms of the cell. So we have an adult human has on the order of 100 trillion cells. And each of the cells, most of the cells have mitochondria in them. And the mitochondria has a little bit of DNA, just a very small amount of DNA. And I showed what that DNA, if we sequenced it and spelled it out, let me see, it would look like that.
From the nucleus or from the mitochondria?
From the mitochondria. So let's go back to the diagrams. I'm going to get rid of that one. So we have 3.3 billion, roughly, bases in the nucleus. And then we have...
For one cell?
Huh?
For one cell?
Yeah, for one cell, 3.3 billion bases of DNA in the nucleus. And then according to MitoMap, we have 16,569 bases in the mitochondria. And this is like less than 1% of all the DNA. If someone wants to do a calculation here, they could figure out this is just like really minuscule. But this is where all the arguments are regarding the view that Eve lived 6,500 years ago. So I hope that's kind of helpful. So I'm really glad I had the chance to show you the actual sequences here. And
Yeah, I mean, this is
when you go off to school. Oh, by the way, you're talking about where you want to get your PhD. If you have any knack for chemistry, chemistry and computers. Chemistry and computers is always good. Chemistry and computers, always good.
Yeah, I really like cell. I think what I'm gonna be like doing, I think what I'm gonna do is just go on Google, type like a cell, like human cell or animal cell, and I'm just gonna study everything like the nucleus, all the part of the cell.
Okay, so now let's just kind of review. We started with the cell, and what does the cell have? It has all these other parts like the nucleus, the mitochondria. And so there are two places we have DNA in the cell. So where are the two places?
And the two places of what? Sorry?
Where does DNA reside in the cell?
In the nucleus and to the mitochondria,
Correct.
to the ATP. Is it into the Oh, I mean around the ATP.
It's
and
near the ATP synthase. The ATP synthase machines are along this inner membrane here. And then in there, and then you have all these little floating mitochondrial genomes here. This is also something I just noticed. I mean, I never thought of this, but if there are multiple little circles here, that there could actually even be more mitochondrial DNAs than I thought. Oh, well. Now, so you can have multiple mitochondrial DNAs. You can have because you have multiple mitochondria in a cell. So do you remember what numbers I said as far as how many you can have in a cell?
6 in a cell, in the total cell?
Yeah.
Around 2.3 billion.
No. How many mitochondria can you have in a cell?
Oh, around 9.
9 if it's like, say, like close to the embryonic stage, up to 600,000 in other cell types. So there are a variety of cells. I don't have the citation for the 9 figure. That's the one Nathaniel Jeanson gave me. I do have a citation for the 600,000 for mitochondrial number of mitochondria in a single cell. The typical numbers you'll see are from 1 to 2,000, maybe 4,000. But there's some that have huge numbers of mitochondria. So that means you have many mitochondrial genomes even in one cell.
That's really amazing.
And you heard that I used the term heteroplasmy. That means that's when one mitochondria has a different genome than another, just a slight variation. That's what heteroplasmy means.
Yeah, I'm gonna be looking more into the to try to understand more the ATP synthase.
And Standing for Truth, if you want to join, please do. You can ask me. You can actually help me. So if you want to join and weigh in and ask me questions, I can go over with you. But I was just teaching BaptistChad some of the...
call me Will, Sal. Will. Yep.
Some of the basics of cell biology. So you basically touched on college junior level cell biology there. Now,
that's really amazing.
That's just barely enough to kind of understand the creationist issues. But obviously, there's a reason that people study this in their junior year. Typically, what'll happen is someone will have to learn general chemistry, then organic chemistry, then biochemistry. And then when they get to cell biology, that's when they get heavy into all of these details. So I've given you kind of kind of a very quick overview, but now you know what we're talking about. And so let me.
Yeah, I've noted.
I need to type something in the chat.
Yep. Really appreciate it, Sal, that you're teaching me.
Oh, well, maybe we'll have some more lessons because if you don't mind me, if you feel comfortable revealing how old you are.
Oh yeah.
Go ahead.
Yeah. So real quick, I'll just make. I'm 15-year-old. I'm close to my 16. I've been a Christian for around 1 years, looking into God creation, and I'm really excited to learn about everything. And I would like to get a PhD in either physics or biology. So I'm great to be here with Sal and trying to learn a lot, the more that I can, and I really appreciate everything.
Oh, thank you. So I'll call you Will. All right.
Yep.
And so I'm glad you're fascinated by this stuff. And may the Lord guide you where you need to go.
Amen.
And may the Lord guide me to be of service to you.
Mm-hmm.
And it's an honor to be able to serve you. So that's a little bit about the cell biology end. And did you want to talk about specifically about DNA?
Yeah, we can go about DNA and maybe protein because I don't have a
Okay.
clue of what protein does or how they do it or
Okay.
what they do.
We can have you have my email. We'll set up a time to talk about email. So
Amazing.
let me see. All right. So let me look. I'm just looking around here. I'm going to throw away some of my Chrome tabs so I have more just to clear up my desk. And that's what I'm doing right now. And I could think a little more clearly. So, all right. So what do you know about DNA?
What do I know about DNA?
Just tell me, just if someone asks you what's DNA, you don't have to try to get this right. Just
I would
say
say DNA is coding. DNA is coding for protein.
okay. That's one of its
Coding how to form organ, coding how to form stuff in our body. It's basically a schematic of us, So in DNA we could find like how to create organ, how the body is made, all the code for creating a human being.
Thank you for telling me that. That's not completely accurate, but that's what you might find in textbooks. So I don't fault you for that.
Mm-hmm.
And so I'm going to share my screen now, and we'll just go to the wiki and see what it says. Now, so this is DNA, and maybe we'll wrap it up if Standing for Truth doesn't join us. Deoxyribonucleic acid is a molecule composed of two polynucleotide chains that coil around each other to form a double helix carrying genetic instructions for the development, functioning, growth, and reproduction of all known organisms and many viruses. Now, a little qualifier. A lot of the blueprints for what makes us human may not be all in the DNA. The DNA has pretty much all the information for the protein sequences, but there's way more to building a human than getting protein sequences, and that's a very advanced topic. So, what is important here is this is deoxyribonucleic acid is a molecule composed of two polynucleotide chains. So when you go off to college, you're going to learn all of these chemical terms, and that's why just to understand the first sentence, that's already you're getting into about junior level biochemistry. So unfortunately, they described it in terms of biochemistry. Now there are molecules. There are four different molecules. One's called adenine, thymine, cytosine, and guanine. I keep confusing. There's a cysteine amino acid, and then there's a cytosine nucleotide. Anyway, so that's where we get this A, T, C, and G, is the molecule on one of the strands here. It corresponds to a letter. And then I showed you, let me see the mitomap. Do I have the mitomap? Did I lose the mitomap? No, here it is. So when you see a sequence like this, it corresponds to the chemicals on the double helix here, the DNA. Does that make sense?
Yeah.
Okay, great. So then what we're talking about is when there are copy errors in the process of cells duplicating, we call those mutations, and the spelling of the DNA can change. And that's a mutation. And so they change spontaneously because of various chemical considerations. And the rate that they change, we can make estimates as to how far back
They goes to
mitochondrial Eve lived. And so that's what was the discussion tonight.
So you're saying that the DNA, so you know the little DNA, the 4 DNA ATCP, so they can change that code right there. So if something happened over there, like the C could change to a basically.
Say that again, BaptistChad?
Yeah. So the DNA, right? It's kind of what we're seeing right now.
Right.
So if something happened in the DNA into a DNA schematic, could the C or a G could change to A?
Yes.
And that could lead to destructive mutation, right? Because if it's not in the right place, it could destroy something rather than add something.
It could hurt, definitely could hurt. Sometimes the genome can tolerate it. Sometimes the organism can tolerate. Actually, the fact that we're still alive is good evidence that we can tolerate those changes. So everything doesn't fall apart. Just like a car can get wear and tear on its tires, there will come a point where it could be too much and the car is going to blow out its tire. So I'll bring to your attention, do you see here like you have an A that's in capital letters? That means that there are some people with, in that position on the DNA, they have a different letter.
So my mother could have a T there.
It's probably unlikely, but that's why they have, that'd be an interesting question. They could sequence your mother's DNA and then look at yours, your mitochondrial DNA. So yes, males have mitochondrial DNA. They inherit it from their mothers. They can't pass it on to their children. But so there'll be variations there. And over time, more and more variations will accumulate in the genome. So that's pretty much it. I'd like to thank everyone for joining us. Let me do a quick recap.
Yeah, Matt says Matt wanted to join.
I think I want to close this down, but let me do a quick recap. The important thing was we had this VarScan tool that is able to distinguish somatic mutations from germline mutations. That means we can use somatic cells to estimate germline mutation rates. And so this was the paper, and you all can see it. And then also I referenced it from this paper, The Landscape of Germline and Somatic Mitochondrial DNA Mutations in Pediatric Malignancies. So thank you all for joining.
Thank you. So I'll be shooting you an email.